So yesterday morning, I told you about a cell lysis method that is very clean and that we're very excited about, but I didn't tell you anything about how it works because I wanted to reserve this privilege for Harrison Specht, a doctoral student in my laboratory. And Harrison is going to tell you what we think is a promising way of lysing single cells so cleanly that there's nothing to remove.
No, we don't? Yeah, no, I was going to try and talk and distract you while I looked for it some more. There it is. So going last is great because I think I don't have to dwell as much on some of these motivation slides. We've seen some really cool examples of the reasons we want to do single-cell proteomics, the powerful things that it can show us, and a lot of the downsides as well. So I get to go last and show you that, but I think also I'm sort of starting to see the 2020 hindsight, and having made these slides before the conference started, there's now analysis that I definitely want to do before we really get this out there and show this as a final product. But anyways, with that in mind, I'll get started. So what we're going to talk about, what I want to show to you is work that we've been working on in the SLAVOP lab for about a year now. And the title that we've given this talk is a little misleading. We're calling it blank. Automated sample preparation for high-throughput single-cell proteomics. It doesn't like me. Okay.
Too bold of a title.
I know, it's... All right, well, that's the first thing we'll change. And that makes it sound a bit like an engineering platform that we've made, but hopefully the things that I'm going to try and show you are going to be very conceptual. And not at all an engineering task, even though we're all coming from the bioengineering department. So with that in mind, I'll get started. Okay, so here's part of that motivation slide that I feel like now we all have seen. Yep. Cool. So yeah, now this slide does sort of woeful injustice to all the things that we've gotten to hear about so far. So I think we're all very motivated that we want to do single-cell proteomics, we want to do lots of them, and that it can really show us very insightful things about the subpopulations and all of these models of biology and disease that we want to study. And the main task at hand, and I think we can all agree about this, and it's emerging, is that the sample processing and the sample preparation is really what we want to innovate on in order to make this a possibility. So some of the criteria, and this is almost identical to some slides that have been shown before. Fast, automated, robotic, that'd be great. We'd like it to be cheap. We'd like it to be very accessible to biologists everywhere. We don't want this to be a niche technique. And one thing that I think we can all definitely agree on conceptually is that it's got to be done in small volumes. Definitely going to be one of those things that we all are going to be trying to accomplish when we do these single-cell preps. So, again, a slide that I think we've seen a little bit before is that basically some of the ways that we commonly prepare proteomes for analysis by bottom-up mass spectrometry may get in the way, may be harder to do than other ways. I'm going to talk about urea and detergents in particular because there's more to automate there. Maybe that's a hurdle. There's more steps to clean up, certainly, and I personally can't even start to conceptualize how to clean up urea and remove some of these chemicals that we might use to break open and fully solubilize these single cells before we inject it into a mass spectrometer. One technique that we've used that sort of obviates this is the focused acoustic sonication. It's got downsides that, again, we've already gotten to discuss a bit. It's expensive, a little slower. It's getting there. And the volumes that you do have to use are comparatively large to what we'd like to accomplish with the volume reduction. So with this in mind, what can we do? How can we get around this? I think that the way we looked at it, and going back to that Model T example, was really simplicity. Let's start with the simplest approach and see if it works. And that's really kind of what we went back to, almost back to sort of the Stone Age here, with a technique that people have been using in biology for a long time to lyse cells, freezing and heating them. We're going to freeze the cells and heat them and perform lysis that way. It obviates a lot of these non-mass spec compatible reagents and gets around the cleanup problem. Totally obviates it, in fact, if you also include mass spec compatible buffers to adjust the pH and accomplish the digestion from proteins to peptides. This has a number of examples, and I've greatly simplified it here, but we certainly spent a long time iterating over different ways of accomplishing this so that it may look unsophisticated, but we've definitely failed at it many, many times, and we have iteratively reached a place where we can accomplish it much better. So it looks simple, but I think that it is also, I'm not going to call it an engineering feat, but it is also a technique that has become sophisticated through iterative development in our hands. But anyway, so that's the basic idea. It has a couple of advantages. The rapid freeze heat is something that is very amenable to plate sorting. And we'd like to do this, and I'll go back as another step and say that we would like to do this in plate format, and this process, this simpler process, would enable that very easily. So anyways, that's the idea. But it's not something that's used in proteomics, obviously. Is it going to be any good for us? So the question we had to answer is, how does it compare to methods that are well validated by time for protein extraction and processing? So we chose one such protocol that's been used around the world many, many times, namely a processing samples by 6 molar urea, lysing and digest using that protocol. Now we turn to SILAC to allow direct comparison between this technique that we're proposing, and we're going to call this technique for convenience mPOP, for minimal proteomics prep. And we wanted the ability to compare that directly in great fine detail across the whole proteome by using SILAC. Now, for those unfamiliar with SILAC, what this allows us to do, the goal of SILAC, is that we would like to create 2 proteomes, identical proteomes, one that is isotopically different from the other, such that they are chemically identical, but we can differentiate them and, importantly, quantitate them with great accuracy using the mass spectrometer. This is our experimental design. We wanted to start with the same number of cells in each of these conditions to be as fair as possible. We fact sorted to do that cell counting. We did a number of replicates. They are processed separately, combined into the same sample. And because, of course, we have urea in the sample, we stage tipped to clean them up. Stage tipping involves basically removing the urea while eluting the peptides separate from the urea. Here's a hypothetical spectra that we can look at, and oddly enough, chose the same colors as SAM for the light and heavies. And what we see here is, for instance, since I'm going to color code urea in yellow and mPOP in blue, we look at this MS1 spectra of an individual peptide, and we can compare their relative intensities. In this case, we see a higher efficiency of processing of protein and peptide extraction in this in urea as opposed to the mPOP sample for this individual spectra. This is a hypothetical spectra, and of course, looking peptide by peptide is very hard, so let's look at the whole proteomes. Here we plot the distributions of the SILAC ratios across the whole proteomes under a number of different digestion conditions, and also performing label swaps of the SILAC under these 2 different growth medias and processings to control for any artifacts that may have been induced in one culture versus the other. Here we see that across the distribution of the whole proteome, we see a certain amount of variation, but we find that the mPOP samples, when are put in the numerator, they compare very favorably. Now, what we should expect, since this is on the log2 scale, is that should these distributions match very closely, namely the peptide ratios be 1 to 1, we would expect a ratio of 0 on this scale. We find that mPOP is doing at least as good as urea with respect to the cell lysis and protein extraction and digestion as well. But this is a whole distribution of the protein. Something that should come to mind is, okay, great, this looks pretty good. There's some variation. But what about membrane proteins or different compartments of the cell that may be very biased under these two techniques? We chose urea because it's tried and true, it's tested. But what about when we go and look at membrane proteins? What we can do then is take this data and look at subsets of it, mapping the proteins to their gene ontologies and extracting out the different cellular compartments and looking at the ratios then. We did a couple here. We looked at membranes in particular, and we found again that they compare very favorably. I'd be curious to hear more about the different ontologies that may or may not be representative. I'm not sure this is not gene mapping to proteins to gene ontologies is definitely not the most accurate thing you can do, but this is one way that we started to suss out the performance across the different cellular compartments. We can also essentially feed the data into gene set enrichment type analysis, and when we did that, we found that no compartments in particular were differential, essentially, and passed an FDR threshold. So having satisfied ourselves that, with this data, that mPOP is performing pretty well, this free seed lysis that we're proposing is performing pretty well compared to the urea processing. We then said, all right, that's good, but how does it compare when we look at the relative quantitation, something that would be more sensitive to inefficiencies of lysis and digestion? So again, we designed an experiment with that in mind. We thought, all right, let's look at two different but fairly similar cell types and see how reproducibly we can get the SILAC ratios from these two different proteomes across these two different processing conditions. The urea lysis we take as a gold standard. We take these U937 cells and these jerked cells, grow them in two different media conditions, two different SILAC conditions. So one is going to be light and one is going to be heavy. We'll be able to get SILAC ratios reflecting the differences in their proteomes. And we process these separately this time. We process them separately by urea, and then the urea sample we clean up by stage tip. The mPOP sample we process separately, and we do not have to clean up by stage tip. That's our experimental design. But what's that going to look like? Here's one such example when we look at two replicates of a urea processed sample, and we look at the jerked over U937 SILAC ratios on the peptide level. And we say, all right, what's good quantitation look like? We take this to be relatively good quantitation and reproducibility of quantitation from the urea sample. So this is one value. We calculate the Pearson correlation for it. So this is sort of zooming in on one sample, or sorry, two samples, and what that would look like. But let's look across all the replicates that we did under a couple of different digestion conditions. We look at the reproducibility, and basically what we're going to see is that the correlations are overwhelmingly positive. The bottom part of the scale is a ratio of 0.8 on the Pearson correlation, and we find that overwhelmingly the correlations, the two proteomes, the two SILAC ratios of the two proteomes correlate quite well across these different preparation methods. So essentially, if you read into this a little bit more, you'll see that the lower correlations here, 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 here correspond to the most disparate of preparative conditions. Namely, if we look at the bottom left-hand corner, a urea sample digested overnight compared to an mPOP prepared sample digested for 3 hours. But again, these are very high correlations. But as when you show heat maps over, it's kind of easy to trick yourself. So let's look at those same values again, plotted as dots, and let's look at the distribution of how those correlations fall and ask ourselves, all right, are the distributions of correlations for these different processing conditions different? And we find when we perform statistical analysis on that we can find those distributions are not statistically different from one another. So we were satisfied there that essentially the quantitation that we are getting out of the mPOP samples is comparable to the quantitation that we're getting out of these urea process samples. And here we compare the proteomes of some of these, and we find that the mPOP samples almost completely subsume the urea, the proteins, identified and quantified in the mPOP samples almost completely subsume those by the urea samples, and those 248 proteins that we see that are unique to the urea process samples don't seem to be correlating to any specific compartment. Great. So at this point, hopefully what I've tried to describe to you and justify is chassis for the Model T. It's something that we would like to use as part of a single cell preparative technique, and that's what we would like to do. So essentially, we tried to validate it using this process, and having satisfied ourselves, we then have taken it and used it to process single cells. Now, to satisfy ourselves that this is working well and to obviate any artifacts that we may be inducing in the sample prep, we sorted single cells into a randomized plate design, 2 different cell types, the adherent HEC cell and a U cell, across 12 different, into a single plate under 12 columns. Each of these columns represents what will become essentially a set, a scope process of, a set of single cells processed by scope MS. And so then we tried to include empty wells that would be full process replicates in each of these different experiments, such that it would allow us to know what does a failed sorting look like, what does a failed digest look like, and to control for any of labeling, co-isolation artifacts that you can imagine for. So we did this, and we also, with the TMT design across these different sets, we used different block patterning. So we did not use the same TMT label schema for the whole plate. We processed this by scope MS. We did this mPOP type processing in 2 microliters. We did it robotically so that we could add very small amounts of reagent in each step. And we used the scope MS design and experimental procedure to accomplish this for 12 such samples. And here's basically a preview, by no means sort of a proof, but here's what the data showed us when we looked at the principal component loading. So essentially, we took all the data that we got from the single plate. We removed the empty wells, because of course that would result in too much imputation. And then we blinded ourselves to where these, what types of cells these were. We processed them and normalized them across the 12 different sets using a reference channel that is on the similar scale as the single cell intensities. And then we looked at the PCA loadings, calculated the PCA components, and reapplied the cell labels and looked at who was where and who was clustering with who. We find that the carrier channel on this principal loading is closer to the HEC channel, or the HEC cells, and this sort of jives with some of the things we've seen, in that HEC cells are often slightly larger, give more intensity per cell, so that I would assume that this mixed carrier design would be more HEC-like, but that's just sort of supposition at this point. If we do a common type of filtering at this point and look at only the proteins that vary between the two, that vary across the different conditions, by filtering on the variants, we see that we can then separate on the first principal component. This is sort of encouraging at this point, that we're truly seeing some structured signal in our randomized plate design, and that this signal is corresponding to the 2 different cell types that we sorted. So that's a preview of the data. I would really love to hear your thoughts on it, any suggestions on things that we can think about and do. What we did this last week was we transfected cells with a fluorescent protein. We sorted hundreds of them by FACS sorting while also quantifying that fluorescent protein that's been transfected. We'll use that as an additional internal reference sample and process them just like this. I hope to show you that data coming soon. But taking a step back, what we hope this is enabling is really that people can go home and say, all right, maybe I'll try this. Maybe that I can go sort a plate, try it out, see what kind of data we get from this, because it really is very accessible. We're using common plasticware, common reagents. TMT, if you're familiar with it, and you probably already know, is the most expensive reagent, but of course we're labeling very little and use small volumes. And that we've done it in an automated way that hopefully will start to get us thinking about how to automate the full process and really make this a truly high throughput and, dare I say, routine sample processing. And with that, I'd like to thank everyone who did this project. I just got to describe it. So we have the whole Slavov lab, Zach, who did all the awesome cell sorting for us, David, who really built the most concrete LC system ever, and Bogdan, who's been a constant inspiration from the beginning. Here's the team that individually contributed to this project. And with that, I'd like to thank Northeastern and NIH. Any questions?
Very interesting. 2 questions. So when you go back to your correlation matrix of your 18-hour and your 3-hour, your 3 hours look better correlated to each other than the 18 hours to themselves, but yet your same samples between 3 and 8 hours look vastly different, or whatever, vastly different in this scale. Why do you think? Do you have any idea why 3 hours looks like it correlates with each other better than it does with 18 hours, or why the 18 hours doesn't correlate as well?
So you're saying the 18-hour digests between the 2 different conditions. Oh, are you looking at the replications
Like
of the same?
it's hard to tell with the colors, but like
Yeah.
that looks better. Right, and I'm just curious, is something happening at like, are you sampling so little that the correlation looks higher? Like, what are the are there more peptides or proteins in the 18-hour to give you more variance, or is it?
Similar number of peptides were quantified in both of these samples, around 5,000 to 8,000, depending. But I think that then they were processed at different times. So I don't have a concrete hypothesis to give you there.
I mean, what are you guys digesting with now for 3 hours or what?
I think, although, wait, just going back 1 second though, if you eliminate this sample right here.
No, you can't do that.
Okay, well if I.
It's in there. You did the experiment. That's the data.
I know, I made the mistake of showing you. But if we don't eliminate that sample and look at the other ones, you'll find similar agreement in these 4 squares and these 4 squares as you do in these 9.
Okay, and then my other question is that we'll talk about that later.
Yeah.
My other question is that do you see any oxidation differences between the methionines and maybe even cysteines when you do the freeze thaw versus the
You let me
urea?
click on my appendix now, Sam.
I've always wanted to look at your appendix.
Cysteine's poorly quantified. Cysteine's poorly quantified. You can look at the distribution of CVs. We get spotty detection of it. Cysteine-containing peptides, sorry.
Okay.
It's poorly quantified, and the CVs are fairly wretched as compared to the rest of them, which have really good CVs across all the conditions. And
What about methionine oxidation?
haven't looked. I will look, though.
Okay.
Very, very interesting indeed. And did I understand correctly that you sorted the cells in water?
So for the bulk preparations, we sorted 10,000 cells into water.
Into water. And then how long did the cells spend in water? What was
So we tried to keep the water at about 4 degrees, but the process of sorting, I didn't record the time, but the time that it takes to sort 10,000 cells and then get it to the freezer was probably on the order of 3 to 5 minutes.
So not too bad. No, the stress were not too stressed, and the hypertonic stress was not popping up the cells even before you've frozen.
Right.
Okay.
Yeah. So there is a fair amount of sheath fluid that does come along with any sorted cell.
How much material do you think, how much protein do you think you lose to the surfaces?
Yeah, great question.
Okay, can I suggest an experiment?
Sure, love it.
This is an experiment that, having watched Rudy for a long time, this is an experiment Rudy would do, because he's actually kind of an old-fashioned biochemist. Radioactively label your cells, and then see what gets left behind in each of those stages. You could do either I-125 or P32, throw in ATP, P32, and let the kinases insert it into proteins.
Cool. That sounds like a really cool experimental design. Yeah, that would be great.
Yeah, I wouldn't know how to do it, so I'm not going to do it. So
So something that I will say that we've done for the single cell prep, I told you that this here is a gross sort of simplification of what we've tried to do. We did passivate the wells using a very simple peptide mixture, such that we didn't lose single cell material to them. So we'll be describing the full methodology in the coming weeks. We're going to throw it on bioRxiv for everyone. So we're going to try and make it as detailed as possible, and hopefully it'll be quite amenable for people to try.
do you have any, so you're in bioengineering, are there any engineers there that are working on transplantable parts for patients? Because one of the big issues that they deal with is essentially biofouling, and so they try to come up with coatings for these devices so they don't get fouled because the immune system and other things come along and try to stick stuff to it. And because I remember there was a guy at University of Washington, whose name I'm blanking on, like 20 years ago, he came up with a way to coat surfaces so they wouldn't foul. And I think he also coated like Eppendorf tubes and nothing would stick to it. So they might have some ideas on how to better passivate the surface so you don't get anything sticking to it.
We've tried some commercial low-bind tubes, but we found that the contamination was not pretty on the mass spec. So the whole point of this is we'd love to do direct inject to minimize losses. But we tried some of these commercial low-bind tubes and found that the contaminants were just overwhelming.
It was Buddy Ratner.
Cool.
So look up and see what he did.
Yeah. I found when we passivated with a simple peptide mixture, we increased the yield by 2 for a couple of experiments. So.
Now, before I give the microphone, I have a comment to the question that Sam asked. So I think that the difference is there. You can say it in the color map, but that's why I prefer to look at those distributions. So if you actually look at the correlations within each distribution, the difference is very small, and some of that can be just stochastic variation. There isn't.
But you're also saying that that's not significantly.
That's right. So
Pretty close. 0.07 is pretty close to like the arbitrary cutoff anyway. So I mean that one could consider those different, right? I mean I can see a difference.
okay.
Yeah. No, I mean, Sam's absolutely right. I think that we're very close to the cutoff there.
Well, we're just wondering if it would be better to digest for 3 hours, and why would that be? Like, is that better because you're not getting carbamylation overnight?
We included that condition. Well, that is one consideration for the comparison, certainly, the carbamylation from the urea. But I think we did the 3-hour digest sort of looking way down the line because we'd like to process these single cell plates in a day and minimize any losses that might happen through surface absorption overnight over that long period they'd be incubated in there. So we included those samples, and you know what? I should identify the dots by the digestion length. But we included those samples with that in mind, with optimizing for the single cell protocol that we hope to do with it.
I mean, Trips is dead after about 3 hours anyway, so I guess that's what I'm wondering is what's happening further down the road. You don't think so? He's had better trips than I do.
Yeah, I think you have very unhealthy trips.
Really? Okay. That might be true too.
So, very nice work. But I was just wondering why you use fact sorting. So you're not really sorting, as I understand it. It's a means to get single cells, right?
We're using it as a means to get single cells.
Because there are other devices, of which I forgot the name now, is using a piezo mechanism to get really nanodroplets of cells in nanodroplets in microtubule.
Are you're referring to the Wolf cell sorter?
It's not a cell sorter.
Not
It's,
cell
it's
sorter.
kind of a dispensing
We'd love to look
device
into it.
to-
We did this because that was accessible to us. I think that
It
there would be.
seems more gentle to
Yeah,
cells
I mean,
than
they're
fact
flying
sorting.
down into the well. Yeah, I totally agree.
Yeah, just want to comment on the Alexander question, how much they were, what's the timing was to not introduce the shock. And we did play, not for this experiment, before that, we played with the several conditions for the fact sorting, and we found that, we tried to fax them directly in water, we try to do a different type of buffers for the sorting, but we found that the most gentle way of doing things just to sort them a normal PBS buffer, which is natural process for anything you do in a sort machine. First of all, they're not swearing too much on us because we try to give them weird buffers to start from. And then second, since this process was the shortest one from sorting to the freezer, that's why there's almost no way we can see introduction of perturbation of anything, even on the phosphorylation level, because I can speak not for this experiment, for the bulk samples that we compare with this and then see if there any phosphorylation, which is the first thing which will pop up if the cells are in shock. So this was a pretty gentle process to do.
Other questions?
So we just that's that too. So I'll ask this: Does your freeze thaw efficiency change with the number of cells? And the cell type, yeah.
That's a great question. Obviously, we've tested 3 cell types so far, but we wanted to. I guess the ultimate answer before I make up more is basically we haven't tested it. I think that we can imagine, we're hypothesizing so far that any cells you can get into a suspension, it will work well for any mammalian cells, any vertebrate cells that you can get into a suspension, it will work well for. But obviously that remains to be seen and tested. I don't think we're going to be doing neurons with this technique.
We have also used this procedure with fairly large bulk samples, with which we could use another procedure and clean them up, but this is so much easier and simpler that we just prefer it over all the other preps if you have cells that can indeed be lysed as well.
I just want to add, because we kind of doing this process in parallel in 2 labs, that's why just 2 updates. It's been done on the bulk. Second updates, it's been done on 10 cell types. They're both working well.
Really simple question. Harrison, have you done experiments to prove that the freezing is necessary?
So early on, it's a good question.
Or tell me, how many experiments did you do to prove it?
Yeah. So I haven't answered that completely, but I can tell you there was one early on experiment where I forgot to do it. And it seemed to work just fine, but we haven't done it in a controlled way.
Why do you not want freezing? Or why do you?
Oh no, I'm just speculating that it might work beautifully without the freezing.
Okay.
So maybe it could be even minimized more.
Yep.
Make sure without the heating too.
Focused accusing.
I very much like Jeroen's comment, because I think that there is a tremendous opportunity for us to reduce the volume by orders of magnitude and miniaturize. So far, we have done something that is very accessible to everybody. We've tried to keep it accessible. And if we go to a device that is more specialized, it will be good for that device to be made by a commercial company, because I want all of you to be able to use it and beyond. So I think one of the virtues of what we have done is the simplicity and hopefully the ease of wide adoption. But we certainly think that this process can be incorporated into microfluidic devices that use much smaller volumes, at which point it might become important, as Paul is suggesting, to perhaps eliminate the freezing step or don't freeze at -80. To the extent that we do the sorting in 96 well plates, it's incredibly easy to put a whole bunch of those in the freezer. It's not really adding much time to the protocol, but if we are doing this in a microfluidic device, it might be desirable to test how much further we can simplify what we have developed.
Great. Any further questions? Thoughts, comments?
Let me give you the microphone.
Thank you. The question is, you want to avoid the triple to SCP column. So do you have a investigate what's the difference if you do the trapping or not trapping? So
we've analyzed some of the, Bogdan has analyzed some of the master mix samples that we created and diluted down to mimic single cell scope sets via trapping and not trapping. And he gets different results from us when we use a trap. So we don't know what the difference in magnitude and yield is from the trap. So that's sort of, I don't have an answer to that, but I think that we really wanted to simplify the process as much as possible.
Yeah, we have a trouble to do the phosphor sample. You will do a trapping, we lost all the phosphor IDs. But for just peptide, we don't see much loss, so it's a good part. Yeah,
Cool.
Thank you.
Thank you.
Any more questions? I have a question for you. So I have enjoyed this meeting tremendously, and I'm hoping to make this an annual meeting that happens every year and grows. And I will just take a very quick poll for you in terms of the timing of the meeting. Do you prefer that this is a little bit after the ASMS, a little bit before, or at a different time unrelated to ASMS? So for the first option, a little bit before ASMS, anybody thinks that's a good time? No, good, that simplifies things. After ASMS, the way we did it now. Okay. A different time, unrelated to ASMS. That actually seems to win.
I think the last probably question should be split into two sections. Summer and not summer. That would be as well a big
Weekend and non-weekend.
Yeah.
Well, then I'll have to take pictures and then count the hands. It's getting complicated. But I can see that there is quite a bit of interest also to have it at another time that is not related to the SMS. So
When you do it right after
Yeah.
next year do it. Independently, because I know people didn't come because they're burnt out from ASMS.
right, yeah.
5 weeks ago, this sounded like a good idea.
In Miami, yeah.
Yeah. Well, I can certainly see a community beginning to coalesce, and that's wonderful. And to keep it going that way, we have organized some social media websites. We have a LinkedIn group that I'm going to email the link to all of the attendees if you want to join it. It's currently very desolate and empty, but hopefully you'll join, and it's going to grow and help us organize forthcoming events like this conference and other activities that are relevant to the entire community. Now we finished a little bit early, so we can start our coffee break earlier. And then as we return from the coffee break at 4:30, we'll break into discussion groups. And so far, we have the discussion groups from the morning that I've mentioned, and we have a couple of group leaders. So if in the meantime, during the break, you feel like leading a discussion on a particular topic that you care about, that you want to hear the opinion of your colleagues in the community, please bring it up at the beginning of the next session when we will break into discussion groups.